Chapter 21, we'll be looking at DNA and biotechnology. When unwound, DNA measures 50 trillionths of an inch in diameter. Each human cell has approximately 3 billion base pairs, which vary by individual, thus our individualized DNA. DNA stands for deoxyribonucleic acid. It's a double-stranded molecule twisted to form a double helix resembling a ladder. Each side and half of each rung is made from a string of repeating units called nucleotides. Nucleotides are the building blocks for nucleic acids, both DNA and RNA. In DNA, the nucleotide consists of a sugar, deoxyribose, in DNA, a phosphate, and one of the 4 nitrogenous bases listed below. The nitrogenous bases in DNA are adenine, guanine, cytosine, and thymine. Adenine represented with a capital A, guanine with a capital G, cytosine with a capital C, and thymine with a capital T. The rungs of the ladder consist of parallel nitrogenous bases. Bases attach according to the rules of complementary base pairing. Adenine A pairs only with thymine T, cytosine C pairs only with guanine G, and each pair of these bases are held together by hydrogen bonds. Our genetic information is encoded in the exact sequences of bases which vary between us as individuals. For example, if you had a section of DNA, whether it be an inch in length or a foot in length, whatever it might be, if you knew that that specific section of DNA contained, say for example, 20% thymine, and you had to determine what percent of adenine that same section of DNA contained, you would also know that it contained 20% because of the base pairing rules and because DNA is a double helix. Subsequently, you could also determine from that same piece of DNA, if you're looking at a sequencing, since you only know that there are 4 nitrogenous bases in DNA, what the percent of guanine and cytosine also were in that section of DNA. So in the example given previously, if asked if it contained 20% adenine, and you knew it thus would contain 20% thymine based off of the base pairing rules, 20 and 20 of course are 40, so you would have to conclude that the remaining section contained 30% guanine and 30% cytosine because of equaling 100%. This shows the ladder type structure. DNA is illustrated as the double helix and the base pairings. You see on your right the base pairs, the T and the A paired together, the A and the T, the sugars that are located between, and the phosphates that's located between each of the sugars. Genetic information is encoded in the exact sequences of bases. DNA must be copied exactly before both mitotic or meiotic cell divisions. The copying process is DNA replication, and this happens during the S phase of both meiosis and mitosis. DNA replication begins when an enzyme, helicase, breaks the hydrogen bonds that hold together the paired bases of the parent molecule. The two strands of the double helix temporarily separate and unwind. Nitrogenous bases, which are also present inside the cell, are going to pair with these temporarily exposed regions. During DNA replication, DNA polymerase link the sugars and the phosphates back to form the double helix. As each of the new double-stranded DNA molecule forms, it twists back into the original structure of a double helix. There's other enzymes involved in DNA replication, such as an enzyme termed gyrase, which helps to relieve the strain or help to relax the DNA as it's being unwound. DNA is considered and termed semi-conservative replication. In each of the new double-stranded DNA molecules, after DNA is replicated, the semi-conservative replication, you have one original parent strand is saved or conserved, and you have the other daughter strand is new. The process of DNA is relatively foolproof, as there are proofreading abilities or capabilities in DNA. It's important to note that even one nucleotide change will change the messenger RNA, which is in turn going to determine the protein produced, which could lead to disease. Complementary base pairing creates two new DNA molecules that are identical to the parent molecule. The replication process ensures our genetic information is passed accurately from parent cell to daughter cells. Again, this shows the process of DNA replication where it has temporarily been unwound inside the nucleus, where you have the parental strand, the new strand with the base pairing rules being applicated, and once again being linked to reform that double helix. Thus, this is the semi-conservative replication where you end up with one new strand and one parental strand. DNA, you might ask, how does it direct cellular activity? DNA codes for RNA, which stands for ribonucleic acid, which in turn are going to code for proteins. And it's these proteins that play the structural or functional role in a cell. As you recall from Chapter 20, a gene is a segment of DNA that's considered the functional unit of inheritance. This segment of DNA of a gene carries these instructions for producing a particular protein or polypeptide. These are expressed when the protein it codes for is produced. The sequences of bases in DNA determines the sequences of bases in RNA, which in turn is going to determine the sequence of amino acids that's produced into a protein. RNA stands for ribonucleic acid. It differs from DNA in that it is single-stranded. It contains the nitrogenous base uracil and not thymine. It still contains adenine, cytosine, and guanine, and it contains the sugar ribose, not deoxyribose. Protein synthesis occurs in two steps: transcription and translation. Translation, because DNA is located in the nucleus, thus transcription occurs in the nucleus. Translation occurs on ribosomes located in the cytoplasm. So once the DNA is replicated, the transcription occurs and DNA is made into the mRNA molecule. It is transported outside the cell through the nuclear pores into the cytoplasm to ribosomes, from which proteins will be translated. Transcription occurs inside the nucleus, as we previously said, and it is the synthesis of messenger RNA using DNA as its template. Messenger RNA carries the DNA's instructions for synthesizing a protein from the nucleus into the cytoplasm onto the ribosomes. The steps in transcription is that DNA unwinds along its hydrogen bonds. Free RNA nucleotides pair with the complementary DNase pairing, C, cytosine with guanine, C and G, and uracil with adenine. Again, there is no thymine nucleotide for nucleic acid nitrogenous base in messenger RNA, and that's an important part to note, discrepancy between the two. So these are going to pair up along the exposed DNA strand, forming this RNA transcript. The RNA transcript is released from the DNA closes again, and this RNA molecule is going to be transported out of the nucleus through the nuclear pores to the cytoplasm. There's three types of RNA produced. You see the mRNA. Later in the chapter we'll talk about tRNA, which stands for transfer RNA, and there's also a rRNA, which stands for ribosomal RNA. mRNA, messenger RNA, carries the information for synthesis of proteins. The transfer RNA is specialized to bring specific amino acids to where it can be added to a growing polypeptide or protein. And ribosomal RNA or rRNA combines with proteins to form the ribosomes, which are the cellular structure on which protein synthesis occurs. These subunits also are formed in the nucleus and transported to the cytoplasm, where they remain separated except during protein synthesis. A promoter is the term given to a sequence of DNA that starts transcription. RNA polymerase is an enzyme that binds with the promoter. It opens up the DNA helix and aligns and links together the RNA nucleotides. So RNA polymerase actually looks for a specific promoter region for a gene to mark the beginning of that gene. The term enhancers refers to segments of DNA that increase the rate of transcription. There's other sequences of bases that signal for stop sequences. So this shows in step 1 the segment of DNA to be transcribed, where this RNA polymerase has identified the promoter region of that gene, unwinds, nucleotide nitrogenous bases inside the nucleus will pair according to the complementary base rulings, A and U, C and G, and the RNA transcript is going to be released, and this is what will be transported outside the nucleus to the ribosome. There are modifications to messenger RNA that occur before it leaves the nucleus. And there's removal of introns. Introns is a term to describe the intervening sequences. The regions of DNA between the promoter and the stop signal that do not have codes that will be translated into a protein. These introns may be sequences that have accumulated over evolutionary time, such as since we are bipedal, or we walk on 2 legs versus 4, the coccyx, which is at the base of our spinal cord, our tailbone, is a fusion that has occurred that gives strength for an upright position, upright walking. Regions of messenger RNA corresponding to the introns are snipped out by specific enzymes. In other words, they will be removed. These sequences will not be transported out into the cytoplasm, so thus they will not be translated into a protein. Exons, on the other hand, are stands for express sequencing. These are left and will be translated into a protein. So introns will be snipped out before the messenger RNA is sent out to the cytoplasm, whereas the remaining exons will be actually transcribed into the protein. Exon standing for expressed. And this is an image showing the initial transcription messenger RNA molecule. Up at the top you see exon 1, intron, exon 2, intron, exon 3. Next step down, enzymes are going to cut those intron areas out. Enzymes will splice this section together, and so thus what will be transported out into the cytoplasm to be translated into a protein will be the exons.